Okay, good morning friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Don't forget to subscribe my channel and also please give your valuable comments. So in the last class actually we were discussing about fertilization and we have seen that zygote is formed. Okay, after the zona reaction or cortical reaction, the zygote has been formed and the other sperms are kept away. The genetic material of the sperm and genetic material of the ovum are now fused together, and in the meantime the ovum becomes now the definitive ovum. It becomes the definitive ovum. That means extra pair of the chromosome has been lost. Only one set of chromosome will be there. Now both the sperm and ovum, they double their DNA as soon as they fuse together. 23 from the male side, 23 from the female side is there. As soon as there is only one set from each side, now both will double their DNAs. So what happens now? There is actually now 23. So male portion will have 46 and female also have the 46. That means one extra pair of the chromosomes will be there. But there is only one centromere. But still it is considered as only one set of chromosomes, because the chromosomes are counted by their centromere, not by the number of chromosomes. Number of chromosomes are more here, but centromere is only one. So one extra pair is formed on each chromosome. Okay, one extra pair is formed. So now there are 23 chromosomes with the double genetic materials. Okay, see, understand this point very clearly. There is no 46 chromosomes. Okay, but chromosomes are only 23 on each side. On each side there is only 23, but in the same centromere another set of chromosomes are formed. Doubling is happening. Okay, so therefore if I say chromosome number 15, chromosome number 15 has got one centromere. Centromere is only one, but along the same centromere now there is formation of, attached to that, formation of one more set of chromosome number 15. Okay, so therefore now there are 23 chromosomes with the double genetic materials. There is no 46 chromosomes if I talk about sperm. Similarly with the ovum. Now what happens? Therefore it is called as 23 to n. 23 to n. Now they are ready for the next generation. Now they are ready for the next generation, and when they are handling the genetic material, handling means when they are having this double genetic material within them, okay, male and female side, both oocyte and sperm. So that is called as growth of the nuclei. That is called as growth of the nuclei. Now this nucleus, which actually contains double genetic materials on each chromosome, so that is called as a male and female pronucleus. Now this nucleus is called as pronucleus. In the male side it is called as a, for the sperm it is called as a male pronucleus, for the female it is called as female pronucleus. Now what happens? Now they will come together. Now for 23 of the male, 23 of the female, they come together. Actually they contain the double materials. Okay, so now they come together to make a new person, isn't it? Now their responsibility, so they have to produce a new person. So both will now undress their membrane. Nuclear membrane will be undressed, and they will now, the nuclear membrane will disappear now, which is so far binding them. So now they come together. They come together and membrane will go into disappears. So now there will be intermingling of the pronucleus of the male and pronucleus of the female. So now again they are arranged in the spindle fashion. Spindle fashion arrangement is important for the cell division, we know that. Okay, so now these are arranged in the spindle. Now all the chromosomes they separate. Okay, all the 23 they separate. The doubling, that also separate. For example, chromosome number 15, it is doubled now in the male side. It is also double in the female side. So now this single and double on the both sides, it will going to separate. So they will spread everywhere. Okay, so totally they are free now. Randomly they are spreading. So now they are arranged. Then again they come back. Now they are arranged in the spindle fashion, and these chromosomes will be decorated in the spindles. Okay, now 15 will come on the 15. Similarly, one more 15 will come with another 15 chromosome or, okay, genetic material. So like that, what happens after this intermingling? So there will be formation of two set of chromatids. Okay, so this newly spindle shaped fashion, arranged in a spindle fashion, these chromosomes, so they are called as chromatids. Now we have got two set of chromatids. One copy of each chromosome will go to one end and other to the other end, and a deep cleft come in between, so there will be formation of the two cells. So this is how the zygote will now divide into two. The first division takes place. So this is how the formation of the zygote takes place and the first division takes place. These are the different steps in the formation of the fertilization. What is the main purpose of fertilization? The main purpose of fertilization is establishment of diploid number of chromosomes, isn't it? Male also contains haploid number, female also after secondary meiotic division it contains diploid number. Okay, so now that's haploid number. So this haploid and haploid should come together and there should be diploid number. That is one important thing. Okay, then second important purpose is determination of the sex of the new individual. Okay, so haploid from the mother side, haploid from the father side, so it helps, they come together. Now there is a decision of the sex. If the haploid which is coming from the father side is a sex chromosome present in it, 23 pairs, 22 plus 1 X or Y. If it is X, it combines with the X of the female also, the gender will be female. Suppose the male portion containing Y and female is always XX, male is XY. So if the male contains Y, then the sex of the child will be male. So this is second important purpose of fertilization. Third one is metabolic activation of the ovum start and there will be initiation of the cleavage. Okay, so metabolic activation of this particular ovum is also possible only after the entry of the sperm inside the inside. Otherwise sperm remains silent. See, if the sperm is not coming, the ovum will undergo destruction after 24 hours. If the sperm is coming, therefore the future things are taking place. Therefore what Ayurveda says, in the basic itself they says, that is Prakriti is Avyakta Prakriti. It's Avyakta Prakriti and it contains all the three gunas. The three gunas are present in the subtle form, in the inert form. When once the Purusha come and enter inside the Prakriti, the fluctuation in the Sattva Guna Tama, Sattva Raja and Tama of the Prakriti will take place, and as a result the formation of the universe starts. So this is what Sankhya Darshana says. Almost similar things are witnessed here also. So with this we conclude the fertilization, and this is in detail about the fertilization in humans. Thank you very much. We will meet again with another class. In the next series of the classes we will go with the fetal development. Thank you very much.